Thank you for watching. I'm Kyohei Yamashita from Tokyo University of Science. This presentation is about a non-invasive cell viability assay using natural food pigments. In dye exclusion test that stains only dead cells, it was found that the toxic synthetic dye can be replaced with the natural monoscous pigment. The results of this research are reported in the paper and patent shown here. Then, let's get right into the main subject. Background and purpose of this research. There is a need for a safe and inexpensive method for viability assay in order to evaluate efficacy and toxicity in basic research of drug discovery and biology science. Dye exclusion test (DET) is widely used in viability assay. Trypan blue and methylene blue, typical synthetic dyes, are used for DET. However, conventional synthetic dyes are toxic to cells and the human body, and thus can determine dead cells but also kill live cells. Samples after DET cannot be used. To solve these problems, we devised to replace highly toxic synthetic dyes with safe and inexpensive natural food pigments. The advantages of this method will be compared with the conventional method. As conventional methods to distinguish between live and dead cells in culture, the following are known. In the exclusion test (DET), cells stained with a dye such as trypan blue is judged as dead cells. In colony formation assay, the number of live cells is evaluated by the number of colonies formed on an agar culture medium. After an inoculation of diluted cell culture and following definite time of culture. In enzyme activity assay, enzymatic reaction of enzymes in living cells or enzymes leaking out of dead cells are used for viability assay. In flow cytometry analysis, dead cells are labeled with a fluorescent dye and detected by fluorescence flow cytometry. However, these methods have drawbacks such as requiring specialized techniques and equipment, damaging cells, and inability to perform in situ measurement in the cultivation process. Therefore, in order to solve these problems, in this research, we propose a cell viability assay using natural edible pigments as the dye exclusion test in the above-mentioned method. Let me explain the principle of dye exclusion test (DET). This is the same principle that salted plums are stained. Living cells are enclosed by the cell membrane, so pigments cannot enter the cells. The photo shows a living green alga, Euglena. On the other hand, dead cells have damaged cell membranes. The pigment invades the cell and binds to intracellular structures, which stain the cell. This is a video of adding purple sweet potato pigment to a cell suspension of unicellular motile green alga Euglena. Living cells are unstained, green, and motile. Dead cells stain red, purple, and are non-motile. It is about the problem of DET. Conventional synthetic dyes are toxic to cells and the human body, so they can stain dead cells, but they also kill live cells. Then, the cells after testing cannot be used. In addition, long-term time-lapse observation requires samples for viability assay at each elapsed time. Making a large number of samples is expensive. Keeping many samples under the same condition is difficult. As a result, the test becomes less reliable. Therefore, in order to perform DET safely, inexpensively, and non-invasively, conventional synthetic dyes are replaced with food pigments. We examined 10 typical natural food pigments to find molluscous and anthocyanin pigment, purple sweet potato, and red cabbage works as a good non-invasive viability indicator of DET for green alga Euglena, protozoa paramecium, human breast cancer MCF-7, among other cells. In particular, molluscous pigment was able to stain various cells of various species with different cell structures. A characteristic of molluscous pigment, different from other natural pigments, is strong staining of proteins. Since cells have proteins generally, it is thought that cells of various species were stained with the molluscous pigment. Therefore, it was suggested that high affinity for the protein is a more suitable dye condition for DET. This shows the result of viability assay with molluscous pigment on cells of various species. The top row of the table shows live cells, which are unstained by pigment. The bottom row shows microwave-treated dead cells stained with molluscous pigment. In this way, molluscous pigment can vividly stain dead cells of various species with different structures. Human breast cancer cells, the subject of this presentation, are no exception. Now, let's move on to the experiment. In experiment 1, we compared the staining ability of molluscous pigment and synthetic dye. Cell death of MCF-7 cells was induced by microwave exposure. After discarding the medium, dead cells were treated with these molluscous pigment concentrations, or 0.1% trypan blue, for 20 minutes, and then washed with PBS. Cells treated with no pigment were used as the control. Cells on the bottom of the dish were observed and photographed with a bright field microscope. Here, The monascus pigment concentration indicated in this presentation means the concentration as a food coloring product. Here is the result. Concentration of monascus pigment above 0.4% give visible staining. 5% monascus pigment stains as strongly as 1% trypan. However, in experiments using live cells, 0.6% or more induced morphological changes in cells. All dead cells were stained within 10 minutes. From the above, it can be said that the monascus pigment concentration that enables non-invasive viability assay is 0.4%. Next, in order to confirm whether the monascus pigment has chemical resistance, a viability assay was performed in the presence of the anti-cancer drug cisplatin. Live MCF-7 cells were treated with 1% or 5% monascus pigment concentration in the presence or absence of cisplatin for 14 hours. After the cells on the bottom of the dish were washed with PBS, the cells were observed and photographed under bright field microscope. Here is the result. These are pictures of cells on the bottom of dishes. Although not shown here, floating dead cells were stained at both 1% and 5% monascus pigment concentrations, with or without cisplatin, leaving MCF-7 attached to the bottom of the dish, whereas dead cells tended to float away from it, regardless of the presence or absence of cisplatin. Control sample containing no pigment and the sample with 1% monascus pigment showed very few floating cells, and 5% monascus pigment showed many floating cells. Although data are not shown regarding cell staining, in 24 hours and 48 hours, 1% monascus pigment stained only floating cells, and 5% monascus pigment stained both cells on the dish and floating cells, regardless of the presence or absence of cisplatin. Therefore, it was confirmed that monascus pigment was not affected by the presence of cisplatin for 48 hours and stably functions as a reagent of viability assay. High concentration of monascus pigment were toxic to MCF-7, as almost all cells were stained with 5% monascus pigment in the presence or absence of cisplatin. Next, the WST8 assay was performed as a cell toxicity assay to confirm the non-invasiveness of monascus pigment. The water-soluble tetrazolium salt WST8 is used in cell proliferation and cytotoxicity assays. When WST8 is reduced, WST8 produces water-soluble formazan with a maximum absorption around 460 nanometer. WST8 is reduced by NADH produced by dehydrogenase in the cell via 1-methoxy-PMS. Since the absorbance is proportional to the number of cells and the enzymatic activity of the cells, it serves as an indicator of the health of the cells. MCF-7 cells were treated with 0.4% monoscus pigment or 0.1% trypan for 24 hours or 48 hours. Then WST8 was added and samples were incubated for 3 hours. The absorbance of the samples was measured. Here is the result. The left graph shows the result 24 hours after the addition of pigment, and the right graph shows the result 48 hours after the addition of pigment. Cells treated with no pigment were used as control. In both cases, the absorbance of 0.1% trypan was significantly lower than the other samples, suggesting a decrease in cell number or a decrease in enzyme activity. On the other hand, 0.4% monoscus pigment is non-invasive to cells due to its higher absorbance than control. This is conclusion. Monoscus pigment was available as a viability assay reagent among 6 natural pigments and stained all dead cells within 10 minutes. Since monoscus pigment has a high affinity with proteins, which are substances common to living organisms, then it has potential as a highly versatile viability assay reagent. In addition, monoscus pigment can be used in combination with the anti-cancer drug cisplatin, and monoscus pigment showed chemical resistance. Monoscus pigment is minimally invasive and 0.4% monoscus pigment concentration is the appropriate concentration. At 0.6% monoscus pigment, a change in cell morphology was observed. Cellular NADH activity was not suppressed in the WST8 assay, even after 48 hours at 0.4% monoscus pigment concentration. In order to make this method into a kit, we will search for untested natural pigments and staining conditions, and test with many cell types. In addition, we would like to elucidate the mechanism of staining and perform viability assay in combination with advanced equipment. These are references. Thank you so much for your kind attention.